Tole barattoota qaqqaalii baga nagaan gara Aliyi Saayins Tuub chaanalitti nagaan dhuftanii tole ውድ ተማሪዎች እንኳን ወደ አሊይ ሳይንስ ቱብ ቻናል በሰላም መጣችሁ። ዛሬ 110 ኢንዛይም ኤግዛም ኩዌስችን ዊዝ አንሰር ይዤ ቀርባለሁ። ሰው በደንብ እንድታዳመጡና የሆነ ነገር ግንዛቤ ከዚሁ እንድትጨበጡ ማሳሰብ እወዳለሁ። ሰው ዛሬ ጋፍሌን አቀባደስን ያደ ባዮሎጂ ኩታ ኩዳ ተኮ በቆና ሰዲ ኢንዛይም ረት kan xiyyeeffateedha. So our today tutorial is on question that is on enzyme with their answer, that is which makes preparation for tutorial that is for preparation for Ethiopian University exam question tutorial. So don't forget like, subscribe Aliyi Saayins Tuub channel. Like, subscribe yogotan wotan galak suda face nakba one tab like subscribe gochun akas mariyo ta kesanifis share gochun dagatina. So let us start our today tutorial is on this enzyme exam question with answer which contains about 110 questions, that is which is prepared from biology grade 11, also old and new curriculum some together. So let us start our today's tutorial on discussing with their answer the question that is prepared from enzymes, which is makes tutorial for Ethiopia University exam preparations. So let us start. Number 1, what do we call the substance upon which an enzyme acts? A, product. B, substrate. C, activation energy. D, enzyme substrate complex. So the answer should be B, substrate. Question number 2, which factor has a more negative effect on the function of enzyme than the others? A, neutral pH. B, very high temperature. C, optimal amount of salt concentration. D, optimum amount of substrate concentration. So the answer should be B, very high temperature have negative effect on the function of enzyme. Question number 3, in which of the following point does the induced Fit model of enzyme action differ from the log and the key model? A, enzyme lower energy of activation. B, substrate bind at active site of enzyme. C, during reaction and enzyme substrate complex is formed. D, the shape of substrate and active site are complementary. So the answer should be C, which makes induced Fit model different from that of log and the key model. Question number 4, which of the following mechanism do cell use to regulate the enzyme catalyzed reaction in metabolic pathway? A, enzyme denaturation. B, irreversible inhibitions. C, competitive inhibition. D, end product inhibition. So the answer should be D, end product inhibition. Question number 5, which of the following class of enzyme digest carbohydrate? A, amylase. B, lipase. C, protease. D, nucleases. So the answer should be amylases. A, which is digest starch, that is a carbohydrate, a type of carbohydrate. Question number 6, when an enzyme is denatured by heat or extreme pH, which one of the following does it lose? A, tertiary structure. B, secondary structure. C, peptide bond. D, primary structure. So the answer should be A, primary structure would be lost in high pH and heat. Question number 7, in competitive inhibitions, which one of the following factor determines the rate of the inhibition? A, enzyme concentration. B, reaction temperature. C, substrate concentration. D, ratio of inhibitors to enzyme concentration. So the answer should be C, substrate concentration determine that of the competitive inhibition. Question number 8, suppose 25% of molecules of an enzyme are inhibited by a non-competitive inhibitors. Which one of the following would happen if the amount of substrate is increased by 15%? A, the reaction rate would be doubled. B, more enzyme molecule would get inhibited. C, the rate of reaction would decrease by 15%. D, the rate of reaction would remain unchanged. So the answer should be A, the reaction rate would be doubled. Question number 9, which class of enzymes that catalyze the joining of two molecules with concomitant hydrolysis of diphosphate bond in ATP. A. Lysis. B. Isomerases. D. Ligases. D. Hydrolysis. So the answer should be C. Ligases, means that the enzyme that joining two molecules is known as ligases. So the answer should be C. Question number 10. Which of the following pair of molecules are known to have catalytic activity? A. Protein and DNA. C. Protein and RNA. C. Carbohydrate and proteins. D. Lipid and proteins. So the answer should be B. Proteins and RNA. Question number 11. To which class of enzyme do digestive enzyme belong? A. Sterases. B. Transferases. C. Hydrolysis. D. Isomerases. So the answer should be C. Hydrolysis. Question number 12. Which of the following type of enzyme inhibitions can be removed when the end product of metabolic pathway is duplicated? A. Allosteric inhibitions. B. Non-reversible inhibition. C. Competitive inhibition. D. Reversible competitive inhibition. So the answer should be A. Question number 13. What is the function of trypsin enzyme in food processing industry? A. Manufacturing hypoallergic baby food. B. Remove food stains from the common food additive gum. C. Tenderize the meat for cooking. D. Break down cellulose into sugar and ferment it to produce cellulose ethanol. So the answer should be A. Manufacture hypoallergic baby food. That is a function of or in food industry that trypsin in food industry. Question number 14. Which of the following substance has a shape which is similar to that of substrate of an enzyme? A. Cofactor. B. The reaction product. C. Competitive inhibitor. D. An allosteric inhibitions. So the answer should be C. Competitive inhibitors have shape that is similar to substrate of an enzyme. Question number 15. Which one of the following is not a coenzyme? A. Vitamin B complex. B. Metal ion. C. FAD. D. NAD. So the answer should be B. Metal ion is not coenzyme. It is a prosthetic group. Question number 16. Which one of the following term refers to the number of substrate converted by one enzyme molecule per second at saturated or fully occupied active site? A. Turnover number. B. Enzyme number. C. Substrate number. D. Product number. So the answer should be A. Turnover number. Question number 17. The most complex structure of protein and that held together by hydrogen bond is called A. Primary structure. B. Secondary structure. C. Tertiary structure. D. Quaternary structure. So the answer should be D. Quaternary structure, which is the most complex structure of protein. Question number 18. The structure of protein, that is the three-dimensional arrangement of all amino acids in a single polypeptide chain, is known as A. Primary structure. B. Secondary structure. D. Quaternary structure. So the answer should be C. Tertiary structure is the three-dimensional arrangement of protein structure. Question number 19. Which one of the following is molecules made of globular proteins? A. Enzyme. B. Keratin. C. Collagen. D. Glycogen. So the answer should be A. Enzyme are globular proteins. Question number 20. Which one of the following is molecule is the building block of an enzyme? A. Glucose. B. Amino acid. C. Nucleotides. D. Fatty acid. So the answer should be B. Amino acid is building block of or building unit of an enzyme. Question 21. Arrangement of the following enzyme in ascending order. Their optimum pH and select the correct option, that's A. Salivary amylase. B. Pepsin. C. Pancreatic amylase. A. CAB. B. BAC. C. ABC. D. CBA. So the answer should be B. Means that the first, the most acidic, that is low acidic, means which between 1 to 3 is the pepsin. Then A is salivary amylase, which is around 6 to 7 pH. when pancreatic is about around 8 pH. So the answer should be B. Question number 22. What are the enzymes used for pre-treatment of bowel mass in biofuel industry and for restriction digestion in molecular biology respectively? A. Lignases and nuclease. B. Protease and amylase. C. Manases and cellulase. D. Beta-glucanase and rennin. So the answer should be A. Lignases and nuclease. Question number 23. Which one of the following is incorrectly matched enzyme with their function? A. Amylase remove starch. B. Cellulase remove protein. C. Lipase remove fat and oil stains. D. Pectinase clarify fruit juice. So the answer should be B is an correct one. Cellulase remove cellulose material, not protein. Question number 24. Which one of the following is mismatched enzyme with their function again? A. Papain tenderize meat for cooking. B. Rennin hydrolyze protein in manufacturing cheese. C. Trypsin manufacture hypoallergic food, baby food. D. Manganese improve beer filtration. So the incorrect one is D. Manganese is not improve beer filtration. It is function of beta-glucanase. Question number 25. What is the function of beta-glucanase enzyme in brewing industry? A. Clarify fruit juice. B. Remove food stains. C. Improve beer filtration. D. Remove fat and oil stains. So the answer should be C. The function of beta-glucanase is improve beer filtration. Question number 26. Which of the following function based at higher optimum pH than the rest? A. Pepsin. B. Salivary amylase. C. Trypsin. D. Enzyme in stomach. So the answer should be B. It have higher temperature, that is about pH that is about 6 to 7. So the answer should be B. Question number 27. Which of the following property of enzyme makes it possible that single enzyme molecule act on the many substrate molecule? A. Enzymes are used over and over again. B. Enzyme lower energy of activation. C. Enzyme are proteins. D. Enzyme are substrate specific. So the answer should be A. Enzymes are used over and over again. Question number 28. Which of the following statement is incorrect? A. All enzymes are not proteins. B. All enzymes are proteins. C. Enzymes are collide in nature. C. Enzymes are inorganic. So the answer should be D. Enzymes are not inorganic. Enzymes are organic catalysts. So also look, A and B. All enzymes are not proteins. Yes, correct. All proteins are enzymes, but not all enzymes are proteins. Don't forget. Question number 29. Among the following, identify the molecule that is an enzyme. A. Starch. B. Cellulase. C. Cellulose. D. Glycogen. So the answer should be B. Cellulase is an enzyme. Question number 30. Which part of human sperm cell contain enzymes that help to penetrate an egg in human female reproductive system? A. Head. B. Midsection. C. Neck. D. Tail. So the answer should be A. Head contains an enzyme that is used to help to penetrate egg. Question number 31. Which of the following enzyme has its optimum pH in acidic range? A. Catalase. B. Pancreatic lipase. C. Salivary amylase. D. Pepsin. So the answer should be D. Pepsin. It is more acidic than the other. Question number 32. What is an alternative name of enzyme pepsin? A. Gastric protease. B. Lipase. C. Trypsin. D. Gastric polymerase. So the answer should be C. Trypsin is an alternative name of pepsin. Question number 33. In an enzymes that contain non-protein organic molecules in addition to protein component, which is protein component is called A. Apoenzyme. B. Coenzyme. C. Holoenzyme. D. Cofactor. So the answer should be D. Cofactor. Question number 34. What which of the following is protein structure and it is description is mismatched? A. Secondary structure, beta plate sheet. B. Primary structure, folded protein chain. C. Secondary structure, alpha helix structure. D. quaternary protein structure, multifolded protein subunit. So the answer should be B. Primary protein structure is not folded, it is amino acid sequence, linear sequence. Question 35. If the ratio of competitive inhibitor molecule is to substrate is molecule is 2 to 9 ratio, the enzyme controlling reaction will be A. 10% activated, B. 20% deactivated, C. 19% activated, D. 29% inhibited. So the answer should be C. 90% 20% is activated when is 20% is inhibited. So the answer is perfectly C. Question number 36. What is the functional group of building block of proteins is called? A. Carboxyl group. B. Ketone group. C. Amino group. D. Aldehyde group. So the answer should be C. Amino group is building block of that proteins and also enzyme. Question number 37. Which of the following belongs to class of enzymes that is known as transferases? A. Esterases. B. Kinases. C. Fructonases. D. Aldolysis. So the answer should be B. Kinases, a group of that of transferases. Question number 38. Which part of human alimentary canal contains digestive enzymes that function at acidic pH? A. Stomach. B. Mouth. C. Esophagus. D. Small intestine. So the answer should be A. Stomach. Question number 39. Which property listed below is not true about enzyme? A. They speed up specific chemical reaction. B. They are used in the reaction they catalyze. C. They are affected by pH and the substrate concentration. D. A small amount can change a large amount of substrate. So the answer should be, the not correct one is B. Enzymes are not used up in the reaction. They can be used over and over again. Question number 40. Which of the following determines the primary structure of polypeptide? A. Hydrogen bonds. B. The code of gene. C. The peptide bond. D. The disulfide bridges. So the answer should be C. Peptide bond determines the structure of primary structure of proteins or polypeptide. Question number 41. How many amino acids are there in all known proteins? A. About 10. B. About 35. C. About 20. D. About 46. So those answer is C. About 20. Question number 42. Which process produce messenger RNA during protein synthesis? A. Translation. B. Replication. C. Mutation. D. Transcription. So the answer should be D. Transcription. Question number 43. What does restriction enzyme do? A. Restriction transcription. B. Cut DNA at specific site. C. Prevent the DNA from replicating. D. Hydrolyze DNA molecule. So the answer should be B. Cut DNA at specific site is the function of restriction enzyme. Question number 44. At which level of structural organization do protein have alpha helix structure? A. Primary structure. B. Secondary structure. C. Tertiary structure. D. Quaternary. So the answer should be B. Secondary structure. Question number 45. Which of the following substance has a shape which is similar to substrate and that blocks the binding of substrate to the active site? A. The reaction product. B. A cofactor or coenzyme. C. Competitive inhibitor. D. An allosteric inhibition. So the answer should be C. Competitive inhibitor. Question number 46. For a biological detergent to effectively remove oil and greasy dirt, which of the following enzyme should do it contain? A. Cellulase. B. Protease. C. Amylase. D. Lipase. So the answer should be D. Lipase. Question number 47. into which follow the following organic compound can lipids, maltase, and sucrase be grouped? A. Hormones. B. Carbohydrate. C. Nucleic acid. D. Enzyme. So the answer should be D. Enzyme. Question number 48. Which of the following pH represents the strongest base? A. 1. B. 7. C. 1,4. D. 1,5. So the answer should be 1,4. Means that when the number of pH increase from 8 to 40, the basic is also increasing. And also when the pH decrease from 6 to 1, the acidic also increase. Question number 49. What are the environmental advantage of using enzyme in industry? A. It makes high production possible with less input of heat. B. It makes high production possible with high input of heat. C. It makes high production possible with emission of more carbon dioxide. D. It helps high production with supply of more heat and emission of more carbon dioxide. So the answer should be A. It makes possible production possible with less input of heat. Means that with less input of heat, the environment pollution also decreased. So the answer is A. Question number 50. When an enzyme is denatured by heat or high extreme pH, which of the following does it lose? A. The peptide bond. B. Primary structure. C. Secondary structure. D. Tertiary structure. So the answer should be D. Tertiary structure. Question number 51. Identify the statement that more correctly explain why each cell contains many different types of enzymes. Enzymes are A. Consumed in the process of catalysis. B. Specific to their substrate. C. Used over and over again. D. Used easily synthesized. So the answer should be B. Enzymes are specific in their substrates. Question number 53. To which category of enzyme do digestive enzyme that break down food substance in human alimentary canal belong? A. Intracellular enzyme. B. Globular enzyme. C. Extracellular enzyme. D. Fibrous enzyme. So the answer should be C. Extracellular enzyme. Means they are digest the food out of the cell. Question number 54. If one keeps on adding substrate to the fixed amount of an enzyme, when does the reaction rate reach a plateau? A. When all substrate is converted to product. B. When all the active site of enzyme are occupied. C. When most enzyme molecules are consumed. D. When much of the product are accumulated. So the answer should be B. When all active site enzyme are occupied. Question number 55. If an enzyme has its optimum activity at neutral pH, to which of the following pH value does this correspond? A. 1,4, pH 1,4, pH 40. B. pH 7, B. C. pH 12, D. pH 4. So the answer should be B. pH 7, which is neutral pH. Question number 56. Which statement is true about enzyme? A. Can be reused. B. Are lipid. C. Change after reaction. D. Increase activation energy. So the answer should be A. Can be reused. Question number 57. Which enzymatic reaction is correct? So the answer should be A. Substrate per enzyme plus enzyme gives enzyme substrate complex, which give product, and again enzyme to use again for other reaction. Question number 58. Which of the following is common characteristics of all enzymes? A. They act inside the cell only. B. They are all proteins. C. They are require activator. D. They operate at any pH. So the answer should be C. Question number 59. What is the formula of chemical kinetic energy of the rate enzyme kinetic energy? Question number 59. What is the formula for rate enzyme kinetic mechanism? So the answer should be D. Substrate enzyme substrate complex, enzyme substrate complex that gives enzyme product and enzyme and plus that product. So the answer is correctly D. Question number 16. An enzyme that carries and transfer methyl group from one compound donor cofactor to another compound acceptor is A. Isomerases, B. Transferases, C. Lysis, D. Ligases. So the answer should be B. Transferases. Question number 61. An inhibitor that binds permanently and blocks the action of an enzyme is called A. Irreversible inhibitor, B. Reversible inhibitor, C. Competitive inhibitor, D. Non-competitive inhibitor. So the answer should be A. Irreversible inhibitor can be bind permanently to the active site of enzyme and blocks the action of enzyme. Question number 62. The two non-protein parts cofactor of an enzyme separate into the firmly and loosely attached to our black speed to enzyme are black speed and specific. A. Simple protein and conjugated protein. B. Vitamin B complex and apoenzyme. C. Prosthetic group and coenzyme. D. Methyl ion and holoenzyme. So the answer should be prosthetic group. C. Prosthetic group and coenzyme are respectively firmly attached and loosely attached to that of holoenzyme. Question number 63. An enzyme that cleavage bonds by other means of rather than that of hydrolysis or oxidation in which two or more substrates are involved in one reaction is A. Hydrolysis, B. Transferases, C. Lysis, D. Oxidoreductase. So the answer should be C. Lysis. Question number 64. An enzyme that hydrolyzes protein will not act upon an starch. This fact indicates that enzymes are A. Catalytic, B. Synthetic, C. Hydrolytic, D. Specific. Means that enzymes that act on protein cannot act on starch. This means they are specific, so the answer should be D. Question number 65. An organic non-protein cofactor which is easily separable from apoenzyme is A. Coenzyme, B. Prosthetic group, C. Apoenzyme, D. Holoenzyme. So the answer should be A. Coenzyme are easily separable from that of apoenzyme. Question number 66. A non-protein cofactor parts that loosely attached to the holoenzyme are usually A. Methyl ion, B. Vitamin B complex, C. Activator, D. Apoenzyme. So the answer should be B. Vitamin B complex. Question number 67. A non-protein cofactor part that firmly attached to the holoenzyme is usually called A. Methyl ion, B. Vitamin B complex, C. Activator, D. Apoenzyme. So the answer should be A. Methyl ion are that of a cofactor that's firmly attached to holoenzyme. Means that methyl ion are example of that of prosthetic group, whereas vitamin B complex is that of coenzyme, example of coenzyme. Question number 68. There are thousands of enzymes in a cell. This indicated which property of an enzyme. A. Enzymes are not consumed in reaction. B. Enzymes are used over and over again. C. Enzymes are specific in reaction. D. Enzymes are used up in reaction. So the answer should be C. Enzymes are specific in the reaction. Question number 69. Which of the following statements concerning enzyme is incorrect? A. Each enzyme has an optimum pH. B. Most enzymes are highly specific. C. Each enzyme has an optimum temperature. D. Most enzymes are made up of RNA. So the incorrect one is D. Most enzymes are made up of not RNA. Most enzymes are made up of proteins. Question number 70. The organic molecules mainly derived from vitamin B complex and methyl ion that essential for activity of enzyme is known as A. Cofactor, B. Coenzyme. D. Apoenzyme. D. Substrate. So the answer should be A. Cofactor. Question number 71. The following terms explain the induced feed model action except one. A. Enzyme lower activation energy. B. Inhibitors bind to enzyme and inactivate its work. C. The active site and the substrate are naturally complementary in nature. D. Binding of substrate produce change in shape of the active site. So the answer should be C. The active site and the substrate naturally complementary. This one is the statement of key and the Locke model. Question number 72. The following feature describes the clusters of biological catalysts except one. A. They are sensitive to the pH, temperature and the pH. B. They do not change the nature of product. C. They are specific. D. They have timeless lifespan. So the answer should be D. Enzymes have a lifespan, not timeless. Question number 73. What stabilize primary structure of protein? A. Ionic bonding. B. Hydrogen bonding. C. Disulfide bridge. D. Peptide bond. So the answer should be D. Peptide bond. Question number 74. If a piece of boiled potato is placed in a test tube containing hydrogen peroxide solution, no reaction observed. This is because boiling A. Denature enzymes. B. Change the pH inside the cell. C. Kills the cell. D. Remove necessary enzyme. So the answer should be it denature enzyme structure. So the answer should be A. Question number 75. The main reason why the rate of reaction increases with temperature initially is A. Time passing allowing more collision to occur. B. Kinetic energy of enzyme and the substrate increase. C. The enzyme become denatured. D. More substrate is produced, therefore enzyme become saturated. So the answer should be B. The kinetic energy of enzyme and the substrate increase, and also the enzyme action also increase. Question number 76. Which of the following is not characteristic of an enzyme? A. It is globular proteins. B. It lower energy of activation. C. It function as catalyst. D. It is converted to product. So the not correct one is that not property of enzyme is D. Enzymes are not converted to product, they are used over and over again. Means they are unchanged at the end of the product. Question number 77. What happens when enzymes become denatured? A. Their active site are changed. B. The rate of reaction increase. C. They can normally catalyze the reaction. D. Bond holding amino acid are unaffected. So the answer should be A. The active site is changed, so the rate of reaction must be decreased, and also the catalyzing enzyme is normally not work, and also the amino acid, also the bonding between amino acid also affected. Question number 78. Molecules that increase the activity of enzyme and molecules that decrease the activity of enzyme are blank space respectively. A. Holoenzyme and a coenzyme. B. Apoenzyme and a holoenzyme. C. Activator and inhibitors. D. Cofactor and a coenzyme. So the answer should be C. Activator and inhibitors. Question number 79. Which of the following can reverse or reduce the allosteric enzyme inhibitions? A. Adding more substrate. B. Depleting the end product. C. Adding more end product. D. Depleting the substrate. So the answer should be B. Depleting the end product can reduce the allosteric enzyme inhibitions. Question number 80. What is an apoenzyme? A. A non-protein cofactor. B. A non-protein molecules. C. An active enzyme. D. A protein molecule. So the answer should be D. A protein molecule. Question number 81. An end product inhibition of the metabolic pathway occurs when A. The last product pathway inhibits the enzyme controlling first reaction. B. The last product of a pathway inhibits the enzyme controlling last reaction. C. The first product of pathway inhibits the enzyme controlling last reaction. C. The first product of pathway inhibits the enzyme controlling first reaction. So the answer should be A. The last product of pathway inhibits the enzyme controlling the first reaction. Question number 82. Which of the following is the first observed discovered enzyme? A. Lactase. B. Diastase. D. Ligase. D. Hydrolase. So the answer should be B. Diastasis. Question number 83. Which of the following statement is incorrect? A. Enzymes are inorganic catalyst. B. Enzymes are thermolabile. C. Enzymes are protein in nature. D. Enzymes are colloidal in nature. So the answer should be A. Enzymes are not inorganic. Enzymes are organic catalyst. Question number 84. Enzymes that have additional binding sites called active site and a regulatory site. A. Enzyme rates. B. Transition state. C. Allosteric enzyme. D. Kinetic energy. So the answer should be C. Allosteric enzyme have additional binding sites that called regulatory site. Question number 85. Which of the following is correct order of malting steeping process? Are A. Germinating, steepening, killing. B. Steeping, germinating, killing. C. Cleaning, germinating, steeping. C. Steeping, cleaning, and germinating. So the answer should be B. Steeping, germinating, and cleaning. Question number 86. The process of cleaning the grains, cereals, and bringing it to life with the water and oxygen by impressing it in a water and air for a specific time period is called A. Germinating, B. Cleaning, C. Steeping, D. Sprouting. So the answer should be C. Steeping. Question number 87. Which of the following mismatched step in additional malting process? A. Steeping means soaking barley grains. B. Germinating, heating the malt. C. Cleaning, heating the malt. D. Germinating, sprouting. So the answer should be B. Germinating, heating the malt. It's not correct. So germination must be sprouting. So the incorrect one is B. Question number 88. The study of biochemistry deals with all aspects except one. A. The synthesis product. B. Immune system. B. Change over state. C. Immune system. D. Expression of the genes. So the answer should be B. Change over state. Okay, question number 89. The application of enzymes are used in the pharmaceutical industry for A. Drugs and antibiotics. B. Starch and fat stain on clothes. C. Sustaining a clean environment. D. Textile and paper making. So the answer should be A. Drugs and antibiotics. Question number 90. Which of the following modifies the protein surface and facilitates intracellular delivery? A. Regulatory enzyme. B. Allosteric enzyme. C. Conformational change. D. Genetic and covalent. So the answer should be D. Genetic and covalent modification. Question number 91. The process of cellular control mechanism in that the end product inhibits enzyme activity is called A. Enzyme inhibition. B. Allosteric enzyme. C. Feedback inhibition. D. Enzyme concentration. So the answer should be C. Feedback inhibition. Question number 92. Which of the following is a control system of enzymatic activity in which enzymes are turned on or off depending on the organism's need? A. Regulatory enzyme. B. Allosteric enzyme. C. Enzyme inhibition. D. Enzyme product. So the answer should be A. Regulatory enzymes. Question number 93. The process of acceleration of a chemical reaction by catalyst is called A. Catalysis. B. Regulation. C. Reversibility. D. Sensitivity. So the answer should be A. Catalysis. Question number 94. Allosteric enzyme contain two binding site called active site or catalytic site and allosteric site or regulatory site for binding blank space and blank space respectively. A. Effector and substrate. B. Inhibitors and activator. C. Substrate and effector. D. Activator and inhibitors. So the answer should be C. Substrate and effector respectively. Question number 95. An enzyme that hydrolyzes protein will not act upon starch. This fact indicates that are A. Catalytic. B. Synthetic. C. Hydrolytic. D. Specific. So the answer should be D. Specific. Question number 96. Which level of protein surface refers to the linear sequence of amino acid? A. Secondary structure. B. Quaternary structure. C. Primary structure. D. Tertiary structure. So the answer should be C. Primary structure. Question number 97. The process of controlling activity of enzyme by activator and inhibitor molecule is called A. Regulations. B. Sensitivity. D. Reversibility. C. Reversibility. D. Catalysis. So the answer should be A. Regulations. Question number 98. The ability to enzymatic biomolecules to catalyze various metabolic such as anabolic and catabolic reaction is known as A. Sensitivity. B. Reversibility. C. Catalysis. D. Regulation. So the answer should be B. Reversibility. Question number 99. The class of enzymes that are catalyze the hydrolysis of various bonds is called A. Hydrolysis. B. Isomerasis. C. Lysis. D. Transferases. So the answer should be A. Hydrolysis. Question number 100. Which of the following is not physical property of enzyme? A. Denaturation. B. Regulation. C. Colloid. D. Precipitation. So the answer should be B. Regulations, which is a chemical properties. Question number 101. Which of the following is not chemical property of enzyme? A. Solubility. B. Sensitivity. C. Reversibility. D. Catalysis. So the physical property solubility. So the answer should be A. Question number 103. The property of enzymes that allows them to be dissolved in water, salt, diluted glycerol, and alcohol causing denaturation is called A. Regulation. B. Sensitivity. C. Solubility. D. Denaturation. So the answer should be C. Denaturation. C. Solubility. Question number 103. Which of the following is tendency of having little or no dialysis across the semi-permeable membrane due to large size and have molecular weights? A. Colloid nature. B. Molecular weight. C. Enzyme activity. D. Biological catalysts. So the answer should be A. Colloidal nature. Question number 104. The separation of enzyme for analysis using different aqueous or ethanol solvent is A. Molecular weight. B. Enzyme precipitation. C. Biological catalyst. D. Enzyme activity. So the answer should be B. Enzyme precipitation. Question number 105. The general catalytic property of an enzyme is A. Enzyme regulation. B. Enzyme specificity. C. Enzyme reversibility. D. Enzyme activity. So the answer should be D. Enzyme activity. Question number 106. Blank space is the minimum amount of energy required for a reactant to be converted to product. A. Enzyme precipitation. B. Enzyme denaturation. C. Enzyme properties. D. Activation energy. So the answer should be D. Activation energy. Question number 107. The process of breaking the intra and intermolecular non-covalent bond that distorts the shape and active site of an enzyme is A. Denaturation. B. Precipitation. C. Reversibility. D. Catalysis. So the answer should be A. Denaturation. Question number 108. The feature of enzymatic activity where an enzyme acts only on a particular substrate is called A. Bond specificity. B. Group specificity. C. Substrate specificity. D. Optical specificity. So the answer should be C. Substrate specificity. Question number 109. Which of the following enzyme convert carbohydrates, proteins, lipid, nucleotide into their monomers respectively? A. Proteins, carbohydrates, lipids, and the nucleus. B. Carbohydrates, proteins, lipids, and the nucleus. C. Lipids, proteins, nucleus, and the carbohydrates. D. Nucleus, carbohydrates, proteins, and the lipids. So the answer should be B. Mean that carbohydrate converted by carbohydrates, protein converted by proteins, lipid converted by lipids, and nucleotide converted by nucleus enzyme into their monomers. Question number 110. What is the conjugated protein part of an enzyme? A. Whole enzyme. B. apoenzyme. C. coenzyme. D. cofactor. So the answer should be A. Whole enzyme. Thank you for watching this video. Don't forget, like, subscribe, share, comment. Aliyi's YouTube channel. Aliyi's YouTube channel. ላይክ ሰብስክራይብ ማድረጉን እንዳትረሱ። አሊይ ሳይንስ ዩቲዩብ ቻናል ላይክ ሰብስክራይብ በማድረግ እኔን የሚለቀውን ነገር ቶሎ ቶሎ እንዲደርሱ ላይክ ሰብስክራይብ በማድረግ ለሌሎችም ሼር አድርጉ። አከሱን በረቶታ ቃሊ ወንተን ገለክሱ ዳፌ አከሰን ገብራ ገውፍ ላይክ ሰብስክራይብ ጎቹ እንደገተና ሄሪዮ ተከሰኒፊስ ሼር ጎቹ እንደገተና ወንተን ገለክሱ ዳፌ አከሰን ብራ ገውፍ ሼር ላይክ ሰብስክራይብ ጎዳ የሮታን ቪዲዮ ቱቶሪያሊ ቪዲዮ ብራ ቀበደ hanga dhufutti yeroo gaarii yaa ta'u yaada qabaattan karaa komentii na barreessaa. Any comments yaada kamiyyuu qabaattan karaa komentii na barreessuu hin dagatinaati. Yeroo itti aan tutoriyaali paartii biraa hanga qabadhee dhufutti yookiin gaafilee biraas qopheessaan jira gara fuulduraatti. Akkasuma tutoriyaalitti baay'ee fuuldura duubaan kan isin dhiyeessu ta'a. Waan hanga yeroo itti aan tutoriyaali biraa qabadhee dhufutti yeroo gaarii yaa ta'u galatooma.